You're about to hear from a very dear friend of ours. We were introduced to him by a wonderful person, Anatoly Evgenyevich Akimov. You remember it. And ever since Pyotr Petrovich Garyaev first came to us, we were amazed and impressed by his research. And more than once, you've asked when Garyaev would visit us again. He's not easy to catch in Moscow. He works in Canada and in other countries and has various laboratories. He has to be here and there at the same time. So after a lot of lucky coincidences, we arranged things with him through Vera Georgievna Yakimova, and he kindly agreed to carve out some very precious time for us and for himself. So before you is the world-renowned scientist Pyotr Petrovich Garyaev. Please pay very close attention to every word. Be extremely attentive to every word. This is advanced science, and we need to draw conclusions for our pedagogy. This is a completely different field altogether. Please welcome him, this remarkable person.
You, as teachers, are dealing with the most important structure of a human being, with its mental structure, which is called the word. It turns out that this word is directly related to our own genetic apparatus. It turns out that thinking is realized through the chromosomal functions of the neurons in the brain. It's an amazing thing, but the neurons of the brain turned out to be the system that most powerfully synthesizes proteins. Proteins, which make up about 80% of our bodies. But in addition, it turns out that this powerful protein synthesis is characteristic of neurons. They have one of the most powerful protein synthesizing systems. The question is, why? These proteins are representations of DNA. Through informational RNA, synthesis occurs with the help of ribosomes. These kinds of protein structures are correlates of consciousness and thinking. Now, at last, we can grab onto that red thread that will lead us to a realistic study of the functions of the brain, the functions of consciousness and thinking, which can't be touched or measured. In this spiritual body, from which our thinking and consciousness are made, seemingly has neither form nor size. As Steiner said, Steiner called this the spiritual body. The body is a purely physical entity, while the spiritual is something that can't really be felt. But nevertheless, this paradoxical spiritual body is the thing that drives us, without which we are simply a conglomeration of muscles, bones, and so on. So when studying DNA, the fundamental and complex genetic structure, the primary genetic structure that actually makes up our biological chromosomes, it was remarkably discovered that the specific sequence of nucleotides in our DNA is surprisingly speech-like in its nature. And a simple, and sometimes not so simple, analysis of the nucleotide sequence showed that these are speech or quasi-speech structures. And we've been working on this, and in Israel, a lot of people are working on it too. This, generally speaking, shocked many scientists, but it's a fact. And so, the correlate, as we see it, of conscious thought, the physical and at the same time mental correlate of conscious thought, are protein phrases. Protein words, protein sentences, which are synthesized in the cerebral cortex, in neurons. Very fast, rapidly changing fractions of these kinds of protein structures. This is characteristic of the genetic apparatus of our neurons. This makes it possible to really dive deep into it. The ideal structure of our thinking, having in our hands a substrate we can work with, and which gives the opportunity, the pragmatic opportunity, essentially to control human consciousness, which, in fact, it has long been doing. You probably know very well about things like neuro-linguistic programming and so on. All of that comes from the same place. In general, Any significant historical figure, above all, had the ability to generate these kinds of verbal wave genes. That is, our speech, whether spoken or written, is a huge conglomerate of wave chromosomes. And everything humanity has written over thousands of years of its existence is a vast encyclopedia of such mental chromosomes, which are very dynamic, very mobile. And yet, in contrast to this, they create the possibility for a person to be stable in the spiritual sense of the word. If a person is unstable, if they are susceptible to verbal viruses, then their mental structure, their spiritual body, falls apart. And after that, the physical body falls apart as well. We know this all too well. When all sorts of false teachings arise, and so on. But when the teaching is correct, it acts like a kind of virus, but a good virus, one that multiplies in our minds. With this virus, this word, we infect other people, including our students. So, specifically, how does it all begin in the genetic parade? It starts with the genetic code. And now I want to show you a certain slide. So this is a small but very important modification of the standard genetic code table. Several Nobel Prizes have already been awarded for this, and it's amazing. Back in the 1960s, when Francis Crick, a Nobel laureate, who, together with James Watson, received the Nobel Prize. As far as I remember, in 1961, for the discovery of the double helix of DNA, the very famous DNA double helix. Since when? Some contradictions have arisen, which Francis Crick himself, as I read in his memoirs, he sort of sidestepped and glossed over. Whether it was intentional or not, on purpose or not, I don't know. It's hard to judge now since he has already passed away. But he wrote a memoir where he stated in black and white that he doesn't see any obvious meaning in the model of the genetic code that he created. So mental constructs and speech constructs actually begin with the table of the genetic code. And when I was a student and a graduate student, it was at the Department of Molecular Biology at the Biology Faculty of Moscow State University. I was surprised to discover it was, as they say in the West, an insight, a revelation, that if you follow the table of the genetic code, which is the very foundation of genetics and biology, the bedrock on which all of biology stands, this table turned out to be contradictory. I remember that I approached my professors, my colleagues, students, and graduate students with surprise. Look, you can see this contradiction, can't you? If you follow the table of the genetic code, then all organisms should die. Because a typical linguistic problem arises there. Homonymy. So there are codon synonyms, meaning those codons that redundantly code for the inclusion of the same amino acid. That's not a big deal. That's just redundancy in genetic information. Nothing to worry about. But there are codons that have homonymy. I won't get into the details. I'll just say that when the protein synthesizing system, sorry for loading you up a bit with molecular biology, but it's important, really important, because this is where what I mentioned comes from. This mental component of our thinking is based on the model of the genetic code, which to this day hasn't been fully understood, not even by its creator, Francis Crick, but no one wanted to be more correct than the Pope. And Francis Crick said that the model has an obvious meaning. Why? So he admitted this before he died. But why is that? Because you find there these typical linguistic constructions, which are characteristic, so to speak, of sentences. These are amine and mess, meaning codons with multiple meanings, which correspond to two identical synonyms. Amine and mess are responsible for synthesis of two different amino acids or stop positions that determine protein synthesis. So if Francis Crick had gone all the way, and had, so to speak, been honest with himself. Right at the very beginning of creating this model, it would have taken genetics in a completely different direction, one that we are now trying to steer genetics toward, namely into mental horizons. And we are proving. We are proving this both experimentally and theoretically, as well as through physics and mathematics, that the genetic apparatus has the ability to think. But of course, this thinking is not the same as at the highest level, at the level of the cerebral cortex. Thinking is fractal in nature, meaning it exists at different scales, and within the framework of the genetic code and the functioning of the genetic apparatus, it is, of course, not comparable to the work of the brain. Nevertheless, the genetic apparatus, roughly speaking, understands and makes sense of things. And as educators, as people who have the ability to construct speech properly, you know this perfectly well. You know that homonyms can only be understood in context. For example, the word kosa or the word klooch has several meanings, and you could assign an exact meaning to these words only in context, that is, within a complete sentence. And only then does it become clear, I emphasize clear, what meaning to assign to the word kosa or the word raid or the word box or the word key. And the ambiguity is resolved, leaving only a single meaning. So it turns out that our genetic apparatus, the one we all come from, we all originate from, our chromosomes, that is, from our DNA, constructs DNA phrases. And in these phrases, you encounter those very same codon homonyms. And in order to choose the correct amino acid and accordingly to produce the correct protein, you need to understand the context of the messenger RNA, which serves as the template for the protein. If there's a mistake, it means death or damage to the biosystem. Just like when we speak and use the wrong words or ambiguous, multi meaning words, homonyms that aren't understood or are misunderstood, you get, so to speak, disinformation effects. And that's how the genetic apparatus works. To understand homonyms, you have to understand the phrase in the messenger RNA. To understand, I want to emphasize this, to understand, to realize, and to make a decision. So genetics is moving into a completely different horizon. That is, if before it was believed, and this has been recognized from the very beginning, that there is a synonymous part in the work of the genetic apparatus, it turns out there's also a homonymous part. But it was ignored and downplayed, starting with the work of Francis Crick himself. And why is that? Because if we admit that there's this kind of linguistic quirk called homonymy, then we have to recognize and understand the work of our genome as an intelligent system. And this, according to materialists in genetics and biology, is not good. It leads to the idea of God. And most of them are materialists. And that's not good. Why it's not good, I don't understand. Personally, to me, God is all of nature, and I don't see anything scary about that. This goes back to Spinoza's time, and a lot of people agree that nature is God. That is, there isn't some person who stands above everything. No, no demiurge. There is nature, not a demiurge. There is a creator of everything. And nature is intelligent. Nature is semantic. If you remember Vasily Vasilovich Nalimov, one of our greatest philosophers, this actually confirms his idea. Just recall his 1989 book, Spontaneous Consciousness. He says that the universe is semantic. So we're coming back again to the classic thesis. In the beginning was the Word, but that's not even an accurate translation. Academician Likhachov said that in the beginning there wasn't the Word, but rather in the beginning there was thought. And thought possesses demiurgic functions. And there are plenty of examples where a person's thought simply works wonders, but everything is built on this. In our world, artificially by our own hands, based on thought, no one argues with that. But there are simple, convincing experiments. Biologists, for example, encounter them quite often. For instance, there was Luther Burbank, an American geneticist, sort of like the American Maturin, right? An incredibly talented scientist who verbally He persuaded. Now, this sounds funny, but it's a classic example and no one disputes it because it's a fact. He convinced a cactus, which had long spines, though the cactus itself was very tasty and people ate it. But those spines got in the way. He tried every selective breeding method to get rid of the spines, but he failed. Then, frustrated, he started talking to the cactus. Now, this sounds funny. He started talking to the plants, to this cactus in particular, and tried to persuade it to get rid of its spines. And over several generations, the spines disappeared. And to this day, that cactus is alive, thriving, and used for food. That's the power of human speech for you. Recently, I had a group of researchers who, on their own initiative, no one forced them, they worked with the plants on their own. And I saw these incredible results when, by mentally picturing the shape of the leaves, the plants, their size, their color, and so on, they would set and program the plants to create a specific leaf shape, like this one, not that one. That's what the human mind is capable of. So let's get back to the genetic code. I'd like to ask for the first slide to be shown, the one I worked on a bit. So take a look here. Here is the famous yin-yang symbol. Here it is, yes, here it is. Now, just a little lower. Can you move it down a bit? Here's the yin-yang right here. Little lower. You see, this is the famous yin-yang symbol, meaning the unity and struggle of opposites. It turns out that in the genetic code, if you rearrange the codons responsible for protein synthesis according to the principle of DNA complementarity, without actually changing anything fundamentally, adenine is complementary to thymine and guanine to cytosine. That's really all you need to do. First, just do that, and second, simply highlight it in red. Codons that are homonyms, and then codons that are synonyms. You see what happens when you do this kind of manipulation? Here are the homonym codons, and here are the synonym codons. So what do we get? We get yin and yang. And this isn't just some random coincidence. The thing is, this is the greatest principle of unity and the struggle of opposites. See, the codons, synonyms in our genetic apparatus, are responsible for the stability of genetic information, for its redundancy. This is synonymy when the same thing is repeated many times under different names. But on the other hand, the genetic apparatus also needs to be flexible, otherwise it wouldn't have that same stability. This flexibility, this multi-meaning semantic load, is carried by the codon homonyms. There you have the linguistic aspect. That's where we all come from. That's your yin and yang, the greatest principle. So there have been studies, as I already mentioned, in the United States, in Israel, and here with us, we've conducted studies where we show that wave processes in DNA molecules and RNA molecules can play the role of systems or subjects that read information. Because right away a question comes up. You say that DNA is text, RNA is text. So what? Are there little people sitting in there who read and tell us what's going on? Yeah. There are strict laws of physics at work there, which, among other things, are reflected in the so-called soliton excitations of the DNA molecule. In other words, these are special waves, stable waves that don't break down, where the nonlinear dispersion network is balanced. They have the ability to read this information from DNA and RNA molecules and transmit it into the intracellular space, and even beyond the boundaries of the biosystem. This is merely a single of its many different dimensions. The work of the genetic apparatus, I repeat, is speech-like in nature. But that's not its only aspect. It turns out there is a second and a third. The second is that our genetic apparatus operates on figurative structures. Speech is also made up of figurative structures, and the biosystem needs information, figurative information, in order to build its four-dimensional structure, that is, its space-time structure. And this is accomplished by what's called holographic memory. Holographic memory, because our genetic apparatus is a hologram. Why is this necessary? It's necessary in order to create certain marking vectors that allow the embryo to orient itself by them, to develop properly so that its arm is in the right place, and so are the leg, the eye, the ear, and so on and so forth. That's what the hologram provides. So official genetics objects to this idea because unfortunately it has already become rigid in all its attributes, despite all the loud claims about great achievements and so on and so forth. Remember the Human Genome Project? $20 billion were poured into it just to analyze the sequence of 3 billion nucleotides in our 46 chromosomes. As a result, after about 15 years of research and spending $20 billion, basically this work was good and necessary, but for us, it wasn't worth that much money. So it was discovered that only 1% of the genes are responsible for protein synthesis, and 99% is junk. Some things have been found in these 99% that help the genetic code responsible for protein synthesis. But the main functions of this terra incognita, well, genetics doesn't understand them, and most importantly, doesn't want to understand them, because it goes against a lot of interests, and that's a long conversation. So these 99% operate on the principles of holography. This is the second aspect of how the genetic apparatus works. After the speech-based one, this one is also figurative. And finally, the third aspect, which is especially interesting, is quantum non-locality, or the teleportation of genetic information. It turns out that not only thoughts can be teleported, which has already been proven by many, even though it's still disputed, but also these entangled states that were formerly predicted by Einstein together with his students Podolsky and Rosen. So people can be in what's called an entangled state, though it's hard to pinpoint. The English word entangle translates into Russian as sputivanya, entanglement, mixing, or intertwining, you could say, right? This is the third aspect of how the genetic apparatus works. So all of this is still being ignored for now, but research is starting to appear, including our own. And the latest book I published today is called Linguistics, the Wave Genome, Theory and Practice. And before that, there was the Wave Genome in 1994, and in 1997, the Wave Genetic Code. And finally, this one right here. Thank you to the people at Kyiv. They helped publish this book because here no one wanted to publish it at all. Moreover, they even allocated $25,000 so that 5,000 copies could be printed. The print run was excellent. I'm grateful to them for that. So now there are works emerging that are trying to understand the genetic code and genetics as a whole from the perspective of quantum physics. And thank God for that. In the beginning, when genetics was just truly taking shape, when the double helix was discovered, physicists got involved. Crick was a physicist, as was Friedenwith-Crick. Max Perutz and other physicists, Franklin, who obtained the X-ray image of DNA, and so on. It all started with physicists. When powerful minds from physics get involved, there's a breakthrough. That's exactly what's happening now. Physicists have joined in. I spent 10 years working at the Lebedev Physical Institute in Molosberg's optics department, trying to convince them that the genetic apparatus operates on two levels. On the material level, which no one denies, and which is mainly involved in protein synthesis, but also on the wave level when our genetic apparatus produces image-based structures. I remember how they laughed at me when I suggested making a DNA laser. A laser based on DNA molecules? Because it's known from the work of Fritz Albert Popp in his school in Germany, I know him personally, that the genetic apparatus, our own genetic apparatus, our chromosomes, and in fact, the chromosomes of all living beings, are structures that emit laser light. So the question was, is it possible to create an artificial DNA laser? I proposed this idea to the optics department, which is a powerful division at FIAN, practically like a separate institute. I tried to convince them for about 5 years, and finally I succeeded. And we created a DNA laser. We pumped DNA and chromosomes in vitro so that they began to emit coherent light. We pumped the yellow larynx, and the emission we got was coherent light around 390 nanometers. That is ultraviolet, blue ultraviolet light. This was published in Quantum Electronics, but it went unnoticed. Quantum Electronics is an excellent, highly rated journal. And 6 years later, 6 years passed, we were still working at that time. In Toronto, a paper by Japanese authors appeared where they did the same work, without citing us, as often happens, and they claimed that they had created a DNA laser. So our work was confirmed. The potential's enormous. If you make such a DNA laser, one that works, that's active, you can load it with any genes and so on. But we took a slightly different approach. We started using our chromosomes with their speech and holographic functions in a different way. And we created equipment that simulates laser equipment, which models these photon laser holographic processes. I'll show you now. Let's flip to the next slide. This is the very first setup. Wenli, back. Is that okay? Here is this modest and unassuming little laser device, which, in memory of Anatoly Gench Akhimov, whom I am profoundly and endlessly grateful to, and Gennady Ivanovich Shipov as well, who is now in Thailand and apparently will never return. These two brilliant scientists. And when I invited them and showed them our effects, our laser, they told me, well, these are torsion fields you have here. There are torsion fields here because this laser, which simulates chromosomal radiation, operates on polarized photons. And what is polarization of a photon? Photon polarization means a change in their spin. So genetic wave information is recorded on the spins of photons. On the spins, on the quantum ones, not on these spins like ours, but on the spins of photons, in other words, on quantum states. So that's what this is about. And this rather modest setup allowed us, first in Moscow, then in Toronto, then in Nizhny Novgorod, and now an independent laboratory has also reproduced it to transmit genetic information read from the pancreas and deliver that information over a distance of 20 kilometers to animals that didn't have a pancreas. We simply poisoned with a special toxin that destroys the pancreas, and the animals died from type 1 diabetes. And in all of them, the pancreas regenerated. Here's a practical application for you. These holographic linguistic potentials of our genetic apparatus, or that of any other higher biosystem. In other words, genetic information can be transmitted over any distance because these are torsion fields, and you can control the recipient biosystem in a positive way. I didn't get that? Well, if you like, then yes, of course. So this laser is a model of how our chromosomes work, and that's at the most basic level, because this is in the red light spectrum, while the main information is actually in the ultraviolet. So you could build a similar setup, but it would be very expensive that would work with blue or ultraviolet quanta. Next slide, please. Here's another example. This is already negative information. So when we regenerated the pancreas, it was a demonstration that, in principle, it's possible to trigger regeneration of any organs and tissues in a person. There are no fundamental prohibitions. In other words, this is a path towards slowing down aging. And here we got, without really expecting it, though we did suspect it, of course, when we damaged the DNA from a plant during the extraction of such pure DNA, there is inevitably a disruption of the structure, including a disruption of the speech structures of the DNA molecules. because all these proteins that are synthesized when reading DNA information are speech structures. And the second disruption, when you change the holograms, that is, when you extract a DNA sample, all the holographic information gets distorted. And what does that lead to? At a distance of 7 kilometers, transmitting this information in all 4 directions, including into the At the Vavilov Institute of General Genetics, where a specialist named Abramov worked, a guy named Volodya, who studied with us at the biology department, a geneticist and expert in plant genetics, specifically Arabidopsis thaliana, he noticed that almost all the plants whose seedlings we exposed to this kind of damaged genetic information, they all mutated. You see, the tall bars represent mutations, and the small red bars are background mutations. So mutagenesis is a deadly process for the plant. In other words, wave genetics is an extremely dangerous thing if you handle it carelessly, and this can have serious consequences. We are careful, and that's why we don't impose. We say, let's study it first and only then implement it. But what does official genetics do? Without understanding the principles of the linguistic nature of the genetic apparatus, the linguistic principle of the genetic code, the protein code, they have imposed technologies on us of genetically modified additives in food products. You know, GMO products, it's a big issue. Right now, environmentalists are fighting against this because it really is Frankenstein food. It would take a long time to explain why, but let me just give you an example. What does it mean, for example, to introduce a foreign protein into the genome of soybeans, a protein that is supposedly beneficial for us, and so on, the product of this gene? So you introduce a foreign gene, a transgene, into soybeans, and you make the soybeans produce some useful protein, like, for example, insulin. Great, sounds good. But what actually happens? When this soy is fed to animals, for example, to rats, they develop cancerous processes, which means there's degeneration in the generations. Newborn rats are affected, and so on and so forth. And there are countless examples like this now. In the United States, all the honeybees died because they fed on fields with transgenic plants collecting nectar there. They are much more sensitive to these things than we are. So here's your first signal, the first loud warning bell that we're heading in the wrong direction, not understanding the linguistic nature of the genetic apparatus. We don't understand, so we end up with genetically modified foods that bring in huge profits. In other words, money rules, gold is king. And we consume all of this, all this unmarked food. This will affect the second and third generations of our children. And something needs to be done about it. This is what happens when we don't understand how things work, how our genetic apparatus functions. Next slide. Now, what we did in Toronto, here's the blood sugar level. First, the baseline blood sugar level. Then, when we poison them with a special toxin, alloxin, which destroys the pancreas, the blood sugar rises. Next, we transmit information at a distance of 20 kilometers or at a distance of 3 meters. Here, 20 kilometers, 3 meters and 1 centimeter. So the blood sugar drops and returns to normal. This means that we regenerated the pancreas. But in Toronto, this work was quickly shut down, and we were sent back to Russia because, to put it bluntly, they just stole our ideas and our equipment. Next slide. These are the guys from Nizhny Novgorod, a group of talented doctors who repeated our experiments. Next slide. This is how the pancreas regenerates. Here is the control, and here is the destroyed pancreas. The islets of Langerhans, they're gone. This is what we got after regeneration. Next slide. Well, this is immunity, specifically the waveform of immunity. These are the tremendous achievements of those guys from Nizhny Novgorod. They figured this out on their own. No one suspected that it was possible to induce wave immunity. But that's a long story. It's a topic all its own. Next slide. Here, academician Garyaev also transmitted genetic information using a laser, but in a slightly different way. This indirectly proves that genetic information really can exist in the form of a field. Next slide. These are wave processes that underlie regeneration, the so-called Fermi pasta ulam recurrence. For us to regenerate, say, an arm, a leg, an ear, or an eye, it means we need to access the memory of how to do it. Lizards do this, salamanders do this, crabs regenerate their claws, and so on. At the core of this is the fundamental process of the Fermi pasta ulam recurrence. And here is reading by DNA molecule solitons. Along this axis is the DNA nucleotide sequence, along this axis is time, and along this axis is amplitude. And you see, the movement of the soliton depends on which section of the DNA this excitation is initiated. So in different sections, the behavior of the soliton is completely different. This is exactly how information is read from DNA molecules, just like from speech structures. Next slide. Well, these are the spectra that contain this high-frequency wave genetic information, which can be transmitted over any distance. Next slide. Next. These are all examples. Here, for example, yeah, and there's nothing to worry about. So here's another one of the practical results we've obtained. So it's not just with rats. Naturally, humans are also subject to the same influences. Here's a woman who's 60 years old. She has no teeth, just dentures. This is her only remaining tooth. So when we were treating her for diabetes, using information taken from the blood of her 10-year-old grandson, who was actively having his teeth pulled, his baby teeth being replaced, her upper and lower jaw started to swell, and she was in shock, and she just couldn't understand what was happening. She got an x-ray of her jaws, and to our surprise, we all discovered that she had grown 3 new teeth. 1, 2, 3. Here are the 3 teeth. And what's more, they grew right into the bone, you see? Right into the bone. This one into the bone, and this one just a little bit. Because she kept wearing her dentures, and they pressed against them and started to move sideways. Here's an example of how it's practically possible. This is a good theory. It's pragmatic. You see, if we use the principles of holography and the linguistic nature of our genetic apparatus, we can achieve things like this. This is the path, the direct path to slowing down aging, so that we don't just live 70 or 80 years, but 200, 300, even 400 years. Or like Noah's sons, who lived for 1,000 years. And maybe even longer, because this program is embedded in our genetic apparatus. But it's locked. For some reason, but it needs to be unlocked, and then we'll have enormous opportunities in our hands. Right now, Russia's population is deteriorating. It's declining. Fewer people are being born and more are dying. What should we do? It means people don't want to have children because you know yourself what kind of atmosphere prevails in Russia. Maybe it's been deliberately set up this way, so to speak. So the only way out of this demographic collapse is to extend people's active lives so that people live for 200, 300, even 400 years. This is possible. And many people in the West are working in this direction, spending billions of dollars, but they're going completely the wrong way, wasting insane amounts of money. The thing is, you just need to understand that the genetic apparatus, I repeat, works on the principles of linguistics, holography, and quantum non-locality, that is, teleportation. Next slide. Oh, this one's hard to see. A bald man, by the way, a talented scientist who worked with Dian Kanjan, or at least used to, came to me, and together we continued this research at Bauman University. And just as we got started, for half a year, we had been working when I got a call from Fortov, the vice president of the Russian Academy of Sciences, a physicist who he is completely removed from genetics and biology. He called the rector of Bauman Moscow State Technical University, and my group was disbanded. This kind of disbandment has already happened 3 times. At least 3 times. That's how the Academy of Sciences treats our field. So with him, you see, after our treatments, literally a month later, his hair started growing back on his bald spot, and we had just read the information from his follicles, his hair follicles. We read the information from the follicles using our quantum biocomputer, which is based on a laser, and there were stem cells in those follicles. That's how we triggered the regeneration process. Here's another practical application of our ideas. Next is Charcot-Marie syndrome, which means immobile legs and muscle degeneration. Here is the before, and a month later, the muscles had already gotten stronger, and the feet started to move. This was at the very beginning of our work, which the big academic institutions have been actively trying to hinder. Next. And this is with a large group of people when our wave matrices, these emissions, we converted them into radio wave form and then recorded them as sound in MP3 format, simply letting people listen to them. This involved several dozen people, and their results improved dramatically. The indicators of acupuncture point conductivity. Here's the before, and this is the after. The statistics are excellent. Next. So this is a very interesting experiment when we intervened in the structure of biological time. This is extremely important. It turns out that biological time, just like any kind of time, has a fractal structure. That is, there are periods of time with a stretched flow and with a compressed flow. And for example, this is very important because if we don't understand the principle of the fractality of biological time, we'll never be able to slow down aging, and we'll never live to be 1,000 years old. So right here, we showed that it's possible to influence the genetic apparatus in this way. Peas. This is just peas. This pea plant is 4 days old, and this one is 28 days old. See? Delayed development, the biological time is different. So what is the fractality of biological time, and how does it work? There are thermophilic bacteria that can withstand 400 degrees Celsius. Everything should fall apart, not to mention the genetic apparatus, so to speak. Everything there should fall apart, but they just live peacefully. Why don't they also live in 2 fractal dimensions of biological time? So they live in our time, at our pace, and that's why you can see them under a microscope, and you can even see their colonies visually. And they also live in stretched out time. That's a different fractal dimension. Temperature is a function of time. If you stretch out time, then you lower the temperature. So by migrating from one fractal dimension of biological time to another, they managed to survive. That's how the earliest organisms survived on planet Earth when it was still hot. This is true for us as well. But this is a big problem for quantum teleportation because it's connected to what's called decoherence. And these cold temporal fractals make it possible for the principles of quantum non-locality to be realized. So next slide. Well, this is also kind of a funny experiment, though it's hard to see. Basically, we managed to slow down the aging of an apple. As you know, apples, if you cut them, they darken quickly because they oxidize, and they spoil quickly. Here's the control apple, already black, and these are the ones where we managed to slow down those processes. That's just one small experiment. There are a huge number of such experiments. These are guys in Israel doing this kind of thing under my influence. Next slide. So this is the so-called phantom effect, which is the subject of a big discussion on the internet. All right, I won't get into that. This is exactly the phantom memory that points to the holographic. Okay, we're back. So this is the DNA phantom effect. That is, if you observe DNA using the method of correlation laser spectroscopy, you see oscillatory dynamics which are visible on the oscilloscope. Before the experiment, there's nothing when we haven't yet introduced the DNA into the laser beam. Then, after we introduce it, we get this kind of oscillatory dynamics. Then we remove everything. You'd think we would return to that original state, but that's not what happens. We continue to detect DNA phantoms. In other words, roughly speaking, after our death, something remains. We don't just disappear. We change. Next, I'll hit. And here is the phantom effect, the classic phantom effect, which we replicated. Adamenko was the first to demonstrate this. So if you cut off part of a leaf, under certain conditions, the cut off part becomes visible. This is called gas discharge visualization. This is precisely a manifestation of the holographic memory of our genetic apparatus. And this holographic memory is manifested in the cerebral cortex, because the cortex operates on holographic principles, but that's a long conversation. Next slide. And these are truly fantastic things we observed when we irradiated and excited DNA molecules with different ranges of light: ultraviolet, infrared, red, and blue. Next slide. As you can see, these kinds of structures fly out of the DNA. It's completely unclear what this even is. It's some kind of information, absolutely extraordinary, that is actually reproduced, and it's unclear what it represents. So our DNA is still an object that needs to be studied, and it contains this kind of quantum wave information, which apparently plays a key role in metabolism. Next slide. Here's another view. This kind of track shoots out from the DNA. In just 1 second, it winds up into this kind of spiral and goes off somewhere. When we saw this, well, we and others who were working on this, there was a whole group of scientists. We were honestly shocked. Next slide. So you turn off the lights, and there's nothing there. You can't see it with your eyes, but the film still picks up this phantom. Next slide. Here's another method. Again, we excite the DNA molecule just with red light, a laser, right? And we photograph it on special film. The DNA was here, and here was the sand. We removed it. There's nothing there. Nothing shows up in the picture. We start processing the digital array using a specific algorithm. People know how to do this. Several rounds of processing, each with a huge amount of information capacity, so to speak. And in the end, we get the DNA phantom. Here it is. This is what remains in the place of the DNA after it's gone. You see? Call it whatever you want. Next slide. Well, this is also one of the effects that happens there. A triple image of the lamp, even though it's a single one. Next slide. This is the same thing, just a little different. So the DNA molecules, when they're excited, yes, they create their own replicas that move to the right. But at the same time, the entire surrounding space is being scanned, and all these devices start producing wave replicas that move to the right. What is this? There's an idea that this is holography on vacuum structures in the spirit of Shipov and Akimov. But all of this needs to be verified. The result is fantastic, and still, I think these studies have a huge future ahead of them. So when you touch the specimen, yes, everything disappears. There are no phantoms at all. Next slide. Next. Well, it's all the same story. And finally, let's move on and look at the last frame. The last frame, please. Next. Next one. Here. This is 2001. We're in the city of Toronto. In a place called Chilbolton in England, an image appears, the so-called response to Carl Sagan. In 1974, some information was sent from the Arecibo telescope in South America to the constellation M950, light years away from us. And there was an image of a human head, our DNA molecules, our solar system, the structure of molecules, DNA, chemical composition, and so on. These kinds of images appeared, it's hard to see here. And a human head. Here's the human head. This is the so-called alien, an extraterrestrial. Next slide. Here's what Carl Sagan sent, among other things, and here, the double helix by Watson and Crick, the classic one. And here's the little human figure with a small head. That's our little head. And here's what those guys sent back. It's impossible to fake because there are such fine details. When Carl Sagan sent this, he specifically included things that couldn't be faked. So this is a real representation of what was sent without any forgery. So their head is much bigger than ours. They're smarter than us. And most importantly, here's the DNA molecule as they depicted it. So the right side is like the Watson-Crick double helix, but the left side is different. So for a long time, people tried to analyze it. Carl Wiegand worked on this. I even wrote him a letter with my own interpretation. They think it's some kind of RNA. It's not RNA. So if it's DNA, it can't function. From my point of view, what kind of warning do we have? That DNA, our DNA, is a quantum oscillator emitting coherent light. This is what we discovered, what Fritz Albert Popp discovered, what we discovered. They are indirectly warning us, ladies and gentlemen, people of Earth, that you are heading down the wrong path with your so-called classical genetics. You are not taking into account the wave functions, the mental functions of DNA molecules, or the speech functions, holographic, quantum, and local. You are driving yourselves into a transgenic dead end, into the apocalypse of transgenic food. So wake up and think about how the genetic apparatus actually works. But no, money determines everything. Markets in Russia, Belarus, Ukraine, and Africa are filled with transgenic products. Americans themselves prefer not to eat transgenic food. So that's a brief overview of wave and linguistic genetics, which is directly connected to how the brain works, to the mental structure of our minds. And I repeat, as teachers, your main working tools are your speech, your verbal constructions, your wave genes, which become part of the treasury of world culture in the form of billions of volumes written by people. This is our chromosomal mental apparatus, which is truly priceless. And most importantly, it must not be distorted. Thank you.